Okay, good afternoon, everyone. I'm Mike Bobola. I'm from the Neurosurgery Department at the University of Washington, and I work closely with Pierre Murad, who could not be here for this meeting. Today I'm going to speak on the use of ultrasound in the therapy of multiple sclerosis. Now, multiple sclerosis is a demyelinating disease. Your nerve cells lose the myelin sheath, and they can't communicate with one another. When your nervous system cannot talk amongst itself, problems occur. Both psychological and physical problems occur. Neurocognitive problems occur. Neural activity promotes myelination. This was the paper that actually prompted us to look further into this. This was when they were using light to activate the neurons, and they saw myelination. The issue, of course, is in this paper, it required a surgery, and they inserted a flashing light into the brain. It worked very well. Unfortunately, you're not going to get FDA approval for putting a flashing light into a human brain. However, this led us to think we could actually use focused ultrasound to activate the brain and develop myelination within the mammalian brain. Focused ultrasound delivery does activate the brain. In our setup, we have EEG monitors to see brain activity, and we put in focused ultrasound at the point indicated here. You see the brain is being activated, and we do multiple cycles of ultrasound as we move forward. Interestingly, the mice that have multiple sclerosis, their brain acted somewhat differently to the ultrasound than normal mice that did not have multiple sclerosis. And upon histological analysis after 15 weeks, we saw myelination of the cells right down where the focused ultrasound was applied. The figure B is just crystal violet to show that we weren't actually damaging the brain with the ultrasound. And the left is a stain for myelin. The myelination caused by the ultrasound was statistically significant, P of 0.02, and a fairly decent T-score. This, of course, was our first go at it to set us up, and this is just an example of one of the brains we looked at. We had a total of 11 mice per cohort. The next steps, more histology, MRI, electron microscopy, and we're going to move from a cuprizome model to an autoimmune model, optimize the focused ultrasound, and we're actually going to include drugs in some of our cohorts. I did it quick. Any questions?
Questions, comments from the audience? I think somebody coming up. Yep.
So were they fed cuprizone throughout the whole treatment, or did you withdraw it? Because they'll remyelinate normally without cuprizone on board.
That is true. And no, we kept the cuprizone going for this particular study. Okay, so the myelination occurred right down where the focused ultrasound was. The contralateral part of the brain did not show the remyelination. And our control mice that were just MS, which were sham treated, and mice that did not receive any cuprizone, came up as appropriate.
So you might want to, MS is not just a cuprizone model. I mean, the EAE would be a much better model because it's an autoimmune disease.
That was my last slide.
Okay.
That's what we're moving to, the autoimmune model.
Okay.
Uh-oh.
Go ahead.
The question about these studies are related with the frequencies that you use in these experiments.
Yes.
What is the selection process of that frequency and the dosage that you use to conduct these studies?
Okay, we used a 1.09 megahertz transducer. We did 20 by 20 microsecond bursts at 1.5 kilohertz. which came out to an ISPTA of 0.06 watts per centimeter squared. We ran through a couple of trials using different frequencies. This was the frequency that appeared to work best. Of course, now that we're changing models, we're going to focus on the ultrasound, change the frequency up to see if something else works a little better, and we're also going to include drugs to see if they work together to further enhance the myelinization. There we go.
So your hypothesis in this regard is the cavitation is the basic mechanism of operation here?
Well, my personal hypothesis is the ultrasound may or may not be preventing demyelinization, but also might be waking up the oligodendrocytes and telling them to stop remyelinate. So one of the two issues that can occur with MS is you're losing the myelin because of an autoimmune attack or some other effect, and the cells that are actually remyelinating things are failing at their job. So it is possible that the ultrasound is stimulating the cells to start making myelin and repairing the damage. And it would be the oligodendrocytes in the brain and the Schwann cells in the periphery.
Thank you very much.
I have two questions about Alzheimer. Can I?
Alzheimer's.
Yeah.
Okay.
Okay, thanks very
So,
Am I going to source? No. Okay.
Go ahead.
so we know today that the plaque in Alzheimer is epiphenomena. It's not the reason of the dementia. And there are people with plaques without any dementia. So what is the reason people working on resolving the plaque, which most of the research now show that Alzheimer is not because of the plaque, it's another process with the tau. This is the first question. And the second question, can you explain how to treat diffuse degenerative disease of the brain with focused ultrasound, which pinpoint treatment or something like that. So it's a diffuse thing.
Okay, thanks. So I think the jury's still, I don't think it's definitive that amyloid is not important for Alzheimer's disease. I think that it's, certainly it's controversial, but I think the amyloid cascade hypothesis is fairly well researched and established. We know that the presence of amyloid triggers a cascade of events that ultimately leads to tau and neurodegeneration. Now, it's not a perfect relationship. There are patients with heavy amyloid burdens that are cognitively intact and vice versa. It's still a target, though, for therapies. And there are several examples of anti-amyloid antibody trials that have failed, but some promising suggestions that targeting amyloid may be of benefit. I agree, and this is where Jürgen's work is so compelling, that we should target more than just amyloid. We should target tau as well, and other neuropathologic features. In terms of and this is the earlier question about Alzheimer's, indeed is a global disease. You have amyloid and tau in many places in the brain. Our view is that one, the idea is not necessarily to target a small region, but to target larger volumes potentially at earlier stages when amyloid and especially tau develop in more stereotypic ways. So if one can intervene at earlier stages when there's less of it, that may be of potential benefit. And also if one is delivering, let's say, a virus or a gene, then that obviates the need for focal treatment.
Can I maybe add one comment? I mean, we worked a lot in this space, basically trying to dissect pathomechanisms in Alzheimer's disease. And we provided support for the amyloid cascade hypothesis, but also had a paper in Cell in 2010, where we showed that when one removes tau, that A-beta is not toxic anymore, at least in animal models. So while A-beta is kind of predicting, let's say by imaging, the conversion of mild cognitive impairment to Alzheimer's disease, tau is more closely correlated with cognitive decline, but still one could envisage that tau has a modulatory role of the toxic effects of A-beta. So it could be that in a patient population which has A-beta deposition, but is not on a fast trajectory to cognitive decline, that tau is somehow altered and thereby modulates the effect of A-beta on cognitive decline.
Any other questions about anything that was discussed in the session? Okay, so if not, we'll close the session. Thanks everybody for the speakers and to the audience for your attention.